In this video, we will continue our discussion of innate immunity. We will begin to examine the components that are involved in the second line of defense. Topics included in this video are the defense components of blood, phagocytosis, and non-phagocytic killing. The second line of defense is activated when pathogens penetrate the first line of defense. The second line of defense includes immune defensive cells, chemicals, and processes that non-specifically act to destroy the pathogen. Many of the components we will encounter originate in the blood or are contained within the blood. We will begin our study of the second line of defense by examining the components of the blood. Blood is a connective tissue composed of non-living matrix with living cells. The plasma is the non-living component of the blood. It is composed of water that contains ions, nutrients, gases, and a variety of proteins which play important roles in immunity. Some of the proteins are involved in the inflammatory reaction and clotting, which, as we will see, are processes that act to prevent infection and blood loss. The proteins responsible for transporting iron in the blood primarily act to transport insoluble iron to cells so it may be recycled, but also act to sequester iron from microorganisms that require it for growth. Iron-binding proteins include transferrin, which binds and transports iron in the plasma, and ferritin, that binds to iron in the liver to allow excess to be stored. Nonetheless, there are some microorganisms that are adapted to acquire their needed iron. Staphylococcus aureus produces iron-binding proteins known as siderophores. The siderophores have a stronger affinity for iron, allowing them to steal iron from the body's transferrins. In response, the body produces another iron-binding protein known as lactoferrin that has an even stronger affinity than the bacteria's protein, thereby allowing the body to steal the iron back from the bacteria. Another group of plasma proteins that are very important in the second line of defense are the complement proteins. We will discuss the function of the complement proteins in more detail. The plasma proteins known as immunoglobulins or antibodies are a part of the third line of defense, the adaptive immunity. The living cells and cell fragments of the blood are known as formed elements. The three types of formed elements include erythrocytes, leukocytes, and platelets. Erythrocytes are the most abundant of the formed elements and serve to transport oxygen and carbon dioxide in the blood. Platelets are cytoplasmic fragments of large cells known as megakaryocytes. Platelets are vital for blood clotting. Leukocytes, also known as white blood cells, act as defensive cells against pathogens. Leukocytes are divided into two groups based on their appearance when examined with a light microscope. Granulocytes are those which have obvious granules present within their cytoplasm, whereas the cytoplasm of agranulocytes lack visible granules. There are three types of granulocytes: basophils, eosinophils, and neutrophils. Basophils contain cytoplasmic granules that stain blue with methylene blue. Basophils are responsible for releasing histamine, which is important in allergic reactions and inflammation. Eosinophils have cytoplasmic granules that stain red or pink when stained with the dye eosin. Eosinophils do perform some phagocytosis. However, they primarily serve to destroy multicellular parasites such as helminths. The cytoplasmic granules of eosinophils contain toxic substances such as cytotoxic proteins and peroxide ions. Eosinophils attach to the outer surface of a parasite and then release the toxic substances so they destroy the parasite by causing lysis of their cells. Neutrophils are the most abundant type of granulocyte. The granules, like those of eosinophils, contain toxic substances used to kill pathogens. Neutrophils have the ability to move out of the blood and into the tissues to the site of an infection. Neutrophils are phagocytic cells that are very active in the initial stages of infection. They are short-lived cells that only live about 8 days and therefore constantly must be replaced. The agranulocytes include lymphocytes and monocytes. Lymphocytes are small cells that contain a single large nucleus that nearly fills the entire cell. Lymphocytes include the natural killer cells, which play a key role in innate immunity by destroying virus-infected cells and tumor cells. B and T lymphocytes are responsible for adaptive immunity. They increase dramatically during the course of an infection. Lymphocytes are not usually phagocytic. They are formed in the bone marrow, and after maturation they are located in the lymphoid tissues and circulate in the blood. Monocytes are large white blood cells that have a single bean-shaped nucleus and lack visible cytoplasmic granules. Monocytes circulating in the blood are not actively phagocytic. Monocytes leave the blood, enter the tissues, and mature into cells known as macrophages, which are very phagocytic. Monocytes have a lifespan of approximately 2 weeks, whereas the lifespan of a macrophage is several months. Macrophages reside within the spleen, lymph nodes, and thymus. They may also be found as wandering macrophages throughout the body or may remain fixed in tissues such as the skin, lung, and nervous system. Phagocytosis is a process in which cells ingest microorganisms or other substances. Cells that are capable of performing phagocytosis are called phagocytes. Phagocytosis begins when cells first migrate to the infected area in response to chemical signals, a process known as positive chemotaxis. Phagocytes respond to a variety of chemicals, including microbial products, components of white blood cells and damaged tissues, defensins, peptides from complement, and chemokines. After reaching the site, the phagocyte must then attach its cell membrane to the surface of the microorganism or other foreign material. The ability to attach to the microorganism is facilitated by the presence of toll-like receptors in the cell membrane of the phagocyte. Toll-like receptors bind to molecules known as pathogen-associated molecular patterns, or PAMPs. PAMPs are molecules found on various bacteria and viruses that are not present in humans. They include molecules such as peptidoglycan, lipopolysaccharide, and flagellin. This process of using toll-like receptors to detect PAMPs is a means of differentiating self from non-self. As we saw in Unit 3, some pathogens escape phagocytosis by the presence of a capsule. The capsule is composed of sugars that are similar to those of the human body and acts to hide the other molecules that are unique to microbes, basically camouflaging the cell and preventing the toll-like receptors from attaching to the microbe. This problem can be overcome through opsonization. Opsonization is a process in which chemicals, known as opsonins, coat the microbe and allow the phagocyte to attach to the opsonins and then permit phagocytosis. Opsonins include molecules such as components of complement and antibodies. After attaching to the microorganism, the phagocyte extends projections of its cell membrane known as pseudopods out and around the microbe to form a phagosome. The membrane of the phagosome has proton pumps that move hydrogen ions to the inside of the phagosome to reduce the pH. To carry out digestion of the microbe, a lysosome merges with the phagosome to form a phagolysosome. The lysosome contains toxic forms of oxygen, nitric oxide, and over 30 enzymes, including lysozyme, lipases, proteases, ribonucleases, and deoxyribonucleases. These molecules act on the microbe to degrade it. The indigestible materials remain within the phagolysosome to form a structure known as a residual body. The residual body moves toward the cytoplasmic membrane so the indigestible materials may be removed from the cell via exocytosis. Some of the microbial components remain attached to the cell membrane of the phagocyte for the process of antigen presentation. We will examine the role of antigen presentation in adaptive immunity. Several cells can kill pathogens without phagocytizing them. Cells capable of non-phagocytic killing include eosinophils, natural killer cells, and neutrophils. Eosinophils, as we have already seen, attach themselves to the surface of parasites and subsequently release toxins that cause the lysis of the parasite cells. Eosinophils can also non-phagocytically kill gram-negative bacteria. The lipopolysaccharide causes the eosinophil to eject its mitochondrial DNA, which combines with proteins that were secreted by the eosinophil to form a physical barrier that acts to trap the bacteria and then kill it. Natural killer cells are a type of lymphocyte that are important in the innate immunity because of their ability to kill virally infected cells or cancer cells. Natural killer cells secrete a chemical called porphyrin that causes the formation of a hole in the cell membrane and then secretes granzyme that causes the cell to undergo apoptosis. Neutrophils utilize two non-phagocytic means to kill pathogens. The first technique is to produce chemicals that act to kill nearby pathogens. Enzymes located in the cell membrane act to add electrons to oxygen to produce peroxide radicals, hydrogen peroxide, and hypochlorite, which is the active component of household bleach. Another enzyme located in the cell membrane has the ability to produce a chemical called nitric oxide. The chemicals kill pathogens that are in close proximity to the neutrophil. Another technique employed by neutrophils is utilized as the neutrophil is worn out and dying. As the nuclear membrane begins to disintegrate, the nuclear proteins and DNA begin to mix with the cytoplasmic proteins and enzymes, forming a neutrophil extracellular trap, known as a net. The net is released as the neutrophil cell membrane breaks down and acts to trap bacteria. The net is capable of trapping gram-negative and gram-positive bacteria. The trapped bacteria are killed by the chemicals in the vicinity that were produced by the neutrophil.